Hi everyone, this is Artem. Welcome to our webinar on generative chemistry at scale. Today we'll share practical lessons learned by Nebius and Quantori in deploying AI-driven workflows from both the platform and scientific tooling perspectives. Let me briefly introduce myself. I'm Artem Elmuratov, Head of Life Science and Healthcare at Nebius. My background is in bioinformatics and computational biology. Over the past decade, I've been involved in building and scaling startups in the space with a focus on developing platforms and tools that support research for genomics, drug discovery, and precision medicine. At Nebius, I lead the efforts to bring high-performance and ready infrastructure to life sciences. Now I would like to invite my colleagues from Quantori, Yuriy and Denis, to introduce themselves.
Hello, everyone. I'm Yuriy Gankin. I'm a Chief Scientific Officer and the co-founder of Quantori, and I have a lot of experience, with 30 years of experience in developing informatics solutions and providing services for the life science and healthcare sector.
Hello, everyone. It's my pleasure and honor to be here. And my name is Denis. I'm a principal chemoinformatics engineer in Quantori with over 10 years experience in academia and 5 years in chemoinformatics and ML engineering. It's my great pleasure to share highlights of the research project we've encountered with Nebius with you today.
Thank you very much, Denis and Yuriy. So here's what we'll cover today. Challenges of scaling generative chemistry workloads, as well as how Nebius enables high-performance computing for life sciences, and real-world use cases and tools from Quantori. Expect practical insights and not just the theory. Here's the agenda for today. We'll speak a bit how our collaboration between Nebius and Quantori started. Then Quantori will introduce themselves, as well as the case of generative chemistry at scale and their model. And we'll speak how you can apply this to your work, as well as the session of questions and answers at the end. A few words about Nebius. Nebius is a vertically integrated cloud platform. We control the full stack from hardware manufacturing to software. We combine the flexibility of hyperscalers with the performance of supercomputers. Our collaboration with NVIDIA ensures all access to optimized GPUs, architectures for Azure and R&D. We are a Nasdaq-listed company backed by deep capital and built for long-term scale. Our engineering-first culture drives constant infrastructure innovation, and as a reference platform NVIDIA cloud partner, our systems are privileged for AI and machine learning workloads, reducing complexity and time to solution for our customers. We serve key life science domains, including drug discovery, for example, accelerated virtual screening of millions of molecules, genomics, first analysis of population and cancer scale sequencing data, health tech, assisted imaging, diagnostics, and clinical decision support. Everything powered by high-performance GPU infrastructure designed for science and research. Our platforms enables training foundational AI models for biology, large-scale inference on cutting-edge models, and accelerated workflows using your preferred tools, all in cost-efficient, scalable, and reliable environment. Now I'll hand it over to the Quantori team, who will walk you through the science and tooling, showcasing how they use this infrastructure to build and deploy generative models for molecule design and drug discovery. Yuriy and Denis, welcome.
Artem, the Nebius team, thank you very much for organizing this webinar and inviting Quantori, and thank you very much for choosing us as a partner. It's my pleasure to introduce our company, Quantori. Quantori is an asset-based informatics services company, and we're focusing exclusively on the healthcare and life sciences. Asset-based meaning that we provide our clients not with just consulting services and development services, but we also provide our clients with proprietary accelerators, platforms, blueprints, and customizable solutions, which we deliver as part of our services. Our main differentiator is in the combination of our deep domain knowledge in healthcare and life science, combined with AI and ML expertise, data analytics, different types, and cloud and software engineering excellence. We provide a wide variety of services ranging from AI machine learning and LLM ops to data science and engineering and laboratory and clinical informatics. Also very strong in everything related to high performance computing. We are proud that our clients think of us as thought leader and a trusted partner. We've published, as Quantori, we've published over 30 peer-reviewed scientific publications since our company was founded. some in collaboration with our clients, some based on our internal research. We were also selected by Everest Analytical Group as a leader in the space of providing information technologies for life sciences. We were selected as a leader in 2024 and also in 2023 as a major contender. We work with 12 out of 15 largest pharma companies based on revenues. We work with multiple innovative and exciting startups and scale-ups. And currently, our size is over 700 people. They have expertise in software engineering, data science, domain specialists, cloud, and all the modern technologies. Approximately 70 people, actually more now, have advanced degrees such as MDs or PhDs in the life science and healthcare. We, as I mentioned before, we develop assets which we pass on to our clients. One of our central assets developed in collaboration with Nebius is our scalable and customizable life science copilot platform that's very flexible and secure platform focused on life sciences primarily, and it supports multiple R&D workflows. We also provide consulting services, and many of our earlier stage clients hire us to develop AI strategy and to develop AI assets. We customize, for example, our AI platforms and accelerators to our clients, brand them for our clients. And so those assets help our clients to increase efficiencies of their workflows and also boost company valuations, as every investor today is looking for AI centricity. And now passing the microphone to my colleague, Denis Sapegin. Denis, the floor is yours.
Thank you very much, Yuriy. And as I already said, it's my pleasure to present the highlights of our recent research project on shape-constrained molecular generation. And without further ado, let's proceed. So what is structure-based molecular generation? Structure-based molecular generation is a modern approach to designing compounds when you have a particular shape in mind, whereas a more commonly employed in drug design, when you need to design a particular molecule to fit inside the void, inside the protein, so-called pocket. It can also be applied to design of very spatially complex molecules in catalysis, macromolecular engineering, material science. And nowadays, machine learning models and other chemoinformatic tools, which are pretty advanced, used by the researchers to tackle this serious and very complicated problem. Nowadays, these tools are mostly used for expanding compound libraries, modifying known scaffolds, or propose new ideas. So it's similar, like you may interact with ChatGPT. You're bouncing off ideas from it to see where didn't you notice something, or what might be a nice transition from your current ideas. In a similar way, from my experience, nowadays in pharma, the machine learning models are used. Although there are a plethora of models already available, but tackling generation based on a shape constraint, so given a shape, you produce a molecule, chemically sound and geometrically feasible molecule that will fit inside the shape or mimic it, it's still not a done thing. And many of our clients are interested in this and looking for ways to tackle it. So what is the way? How can you learn how to create particular structures? Recently, a broad field in generative AI emerged by applying stable diffusion models to molecule generation about 3 years ago. So similar to how stable diffusion models generate images, they can be trained to generate any other object, for example, a molecule, with one particular distinguishable feature. In order to successfully generate a 3D molecule, the denoising operation typically employed in stable diffusion model needs to be invariant according by translation, rotation, and permutation. That means that application of such function to a noised input should be invariant on translation of this noise, on rotation of the noise, and on permutation of the number of atoms. And equivariant diffusion models are great in mimicking and recognizing the molecular geometry and remembering it, because they operate basically on 3D conformance, on atoms in 3D space. And when you feed the model with a 3D conformer, you feed it a lot more than just a chemical structure. You feel potentially you fit it with how energetically variable it is, because you can control which conformers you feed to the model, what are the possible rotations around bonds and what are the distances between atoms. So EGMs learn geometry directly, and they generate chemically valid structures, realistic spatial arrangements, and good coverage of conformational diversities provided by them. Okay, we covered how we can theoretically generate a molecule, but we want to context it on something, and we want to generate something based on our shape constraints. So how do we tell a model what is shape? So that's where shape descriptors come in place. And in order to successfully design and develop a model which will satisfy the production, which will provide you with production grade inference and optimize training efficiency, you would need to choose a proper shape descriptor. They're very important in model performance, inference speed, and training efficiency, because before you will generate your molecules, or when you train your molecules, you will need to create the shape descriptors for all the examples of the training data set. Or if you're doing the inference, you will need to encode this shape into the descriptor and then feed it to a model for further inference. And that basically a common approach is to pick the shape as a voxelized molecular volume, which is pretty clever, and then run it through a shape encoder, either machine learning with trainable weights or something which can take voxelized volume into number of coefficients. And usually such descriptors are very precise, yes, but they're very large in terms of how many values are there. They're quite complex to calculate. When we're talking about encoders which are trained, they need retraining, and most of all, they have low interpretability. So while tackling the task we've encountered to conquer, we decided to follow an interesting approach of simplicity. So we thought that a very interesting physically grounded descriptor which gives a lot of shape inside is the moment of inertia tensor. And if we are more concrete and precise, the principal components of moment of inertia tensor, just free float values to describe the whole shape. It is very fast to compute because it's a basic physics problem. Its rotation are very interpretable. It gives surprisingly competitive precision to application of the voxelized complex encoders, which we'll learn about a little bit later. And most importantly, because we know what it is, we can operate with it. So we can use the properties of moment of inertia for inference tricks, such as in-painting or increasing the model's context window, if I may say. So let's talk about how we trained the model. And the training of the model was made solely possibly by Nebius. Because of the Nebius simple entry point to the architecture, our engineers were able to easily train at scale on 8 NVIDIA H200s, which is about terabyte of GPUs. That's really a lot. And this allowed us to run the training on 1.6 million of ChemBL compounds. ChemBL is many of you may know, is a manually created database of drug-like compounds. And 1.6 million is about 85% of its small molecule subset, which is pretty impressive, at least to me. The one question arises, so maybe you don't know, maybe you do, but in ChemBL you don't have conformers. You have smiles, which are string representation of molecules, so no information-free geometry. So what we used and what Nebius allowed us to do is in each training, we generated these conformers following the algorithms we picked out to generate valid conformers most interesting to us and to our opinion, presenting the training space and chemical space of these conformers, which allowed us to capture all these relationships and distribution of the conformist geometry. The model architecture is presented on the right of the slide, and I will just briefly go through it. So as input, we use a shape. We can use virtually, theoretically, any shape, but in production version, we can use either a ligand, an SDF, PGB, or XYZ file, or a protein pocket shape, or any other shape you want, given that it's properly scaled, in an STL file. So we compute the moment of inertia, generate a batch of samples, and then we supply it to EGM generator that was trained on Nebius platform. And this is where the magic happens. So from the noised inputs, the atoms are aligned in the particular spaces, providing us with a valid geometry. Then to guess the bonds, we use customly trained graph convolutional network. And we use that rather than deterministic pipeline to account for the distribution of chemical features in the target data set. The fine-tuning and retraining of this layer, which is really, really much lighter than the EDM generator itself, allows us to mimic the distribution of target chemical features inside the client's data set, which gives a lot of flexibility here with less value. And finally, the model finishes the structures by running them through deterministic chemoinformatics standardization pipeline, where we constrainedly optimize the geometries, make sure that all the chemical compounds we obtain are valid, and no bad things happen, if I may say. And as a result, the user has an output of a molecule list ranked by shape similarity, which is assessed through intersection of Gaussian molecular volumes, and is able to do whatever he or she wants with it. So run additional chemoinformatics investigations, submit them directly to docking or anything. So what are the performance benchmarks of the model? Currently, we're very proud to say that the model is really fast, and on Nebius H100 GPUs, it may output more than 4 molecules per second, which gives you an ability to generate 50 samples under 12 seconds, which is pretty great. We have achieved maximum shape-to model similarity of more than 99%, which is great. And at the same time, the samples, when generated from scratch, so not conditional generation of the fragment, have very low chemical similarity to the reference, meaning that the variability of the model and basically its ability to generate novel structures, unlike structures, is very high. And final thought is about our ability to match the chemical feature distribution. Is fresher fingerprint distance is a kind of metric that allows you to compare two set of distributions, and the measured distance from generated data set to most commonly employed real data sets of molecules. These are Campbell, PubChem, and Zinc 250K drugs. Is lower than 5, which is, I think, remarkable. So we're able to capture the chemical diversity and mimic real molecular data sets. So what is the use case of this model? So from my personal experience working with our clients, usually these tools, as I've already said, are employed to ideate and test ideas and utilize them in their chemoinformatics workflows. So we think of it as a tool that can be integrated in DMTA lifecycle, or in tools where you can design molecules to provide faster generation of molecules tailored to a specific shape, maybe get some ideas, ideate on something, generate chemically sound, geometrically accurate molecules, and use 3D graph-based representation instead of smiles, which are commonly used in the competitors' models, such as reinvent and Molmim, basically resulting in accelerated time to market for new molecules. So these are a couple of examples what our ML conformer generator can do. And in here you can see that it is perfectly fine capturing this V-shape nature of molecules and outputs pretty decent structures. Even with complex and vivid shapes like crown ethers, you can see that the model can match them, though this is not quite chemically sound, but this was only a trial run. So given that you have thousands of molecules like that, it can filter them out. And even on large molecules, you can see that pretty much the performance is great. I was talking about not only generating the molecules based on molecules, but based on arbitrary shapes. And here's the example of generating some of such molecules based on CLK1 shape, the protein which is employed in mitochondrial oxygenation processes. So here we supplied the model with an STL shape and allowed it to generate molecules that fit in there. For the docking experiments, revealed that some of the molecules were docking quite good, although we understand that outer docking is not a best tool to judge if the compound is really docking to a protein, if it's applicable. But it gives you an idea how it can be utilized. So generally, it can be applied. The model itself can be applied to in silico ideation on the drug targets. So what you can do with our model and with what we have achieved with Nebius. First of all, we are now working on inference as a service and will be available on Nebius Marketplace and other marketplaces shortly. What you can do now is access the model on the GitHub. The inference Python API is free of charge, and the weights of the model are available on Hugging Face under non-commercial research only license. So you can evaluate your model without anything. You just need your own laptop. The model runs on CPU not quite long, so you can evaluate it in there. What we can provide you, if you're interested in application of such workflows in your day-to-day job, we can provide custom training and fine-tuning, adding additional constructs, other shape other than shape, QED, log D, log B, whatever you think of, we can do it. And what we can do if you don't require the model, but you want to, for example, a custom data set, for example, for a certain protein, you want to generate 1,000 targets, which will be chemically sound, cheminformatically filtered. We also can do it thanks to Nebius and our partnership. And last but not least, I would like to demonstrate you the demo app, which is soon be released. And I guess after the webinar, you will have an opportunity to access a beta version, which is free of charge. So here it is. So how it works, and basically this is real generation, will be available and runs on Nebius machine with 1-800. So how it works is you supply it with a mol file. For example, say you can control how many molecules are to be generated and how much variance in number of atoms in the molecules you want. Then we click generate, wait for 10 seconds, I guess. Yeah, and here we go. So you got all the molecules you need here ranked by shape similarity to a reference. You can play around and see how this model, how good the model is, not just trust me, and evaluate if it outputs nice structures that you like or don't. I think that's all.
Thank you very much for that, Yuriy and Denis. And our Quantori team had made a tool available online. You can try molecule generation and get a feel for how generative chemistry can be explored interactively. And we'll share you with the details for it, how to access it. It's a great hands-on way to connect with real outcomes. And you have these QR codes to test it. Thank you very much.
Thank you very much, attendees. Thank you very much, Nebius.
Thank you very much, everybody. Hope to see you.